Okay, yes, so my name's Tonk. I'm a senior professor at the lab. I've been there since 1990. I'm deputy director of our cancer centre. Thank you very much. Did you put the pointer up? Oh, got it, okay. All right, so the problem that we're addressing is a major one, and that is
Give us a second. There we go.
it's still shining on that one.
No, we don't want that dark. Let's try that.
But it's the projection on there that's the problem. You don't see what's going on. We'll just have a look. Okay. There's some interesting stuff in the corner of those slides. You'll have to Okay, so what this actually was, what I was hoping to show you, this is the trend in obesity around the United States. What we're plotting is body mass index. And so you can see back in 1990, it's all these cold colours. Some states didn't even bother to measure body mass index in the population. The rest of them, it was sort of around about 10%. Got worse by 1999. By 2008, you can see all these hot colours here, including in some cases, greater than 30% of the population of some states are effectively obese. And this is a worldwide problem. In 1999, diabetes and its complications account for 30% of US Medicare budget. And the cost of the US is about $135 billion a year at the turn of the century. It's going to get worse. We can solve this problem. We can solve the healthcare crisis in the United States. The second issue is breast cancer, and that is one of the major cancers that affects the population here and worldwide. We're also focusing on a particular form of breast cancer that is characterized by the presence of the oncogene HER2 and is the target for a drug that you may have heard of, Herceptin, which has the disadvantage of being, although very effective, very, very expensive. Okay, the classic approach that's taken the state of the art at the moment is to target what we call signal transduction, the mechanism by which cells respond to their environment, respond to growth factor, hormones, and stresses. And they do it by the reversible addition and removal of phosphate groups to proteins, and this creates a signature in the cell that defines the disease state. Companies are now approaching this through inhibiting one of the enzymes responsible for signal transduction, which are enzymes called kinases that put phosphate groups onto proteins. These are powerful approaches, and they're giving us new ways of treating major diseases. There's a second component of this, and it's the enzymes called the phosphatases, and this is the alternative approach. These enzymes are underexploited, and this is what we focus on. In particular, we focus on an enzyme, a protein tyrosine phosphatase called PTP1B. This was an enzyme I discovered 25 years ago, and it's been one of the focuses of my research ever since. This picture shows you a normal mouse fed a high-fat diet. This is a mouse in which the PTP1B gene has been removed, fed the same high-fat diet. These mice display enhanced sensitivity to insulin and resistance to diet-induced obesity. This tells us that an inhibitor of this enzyme could be a powerful strategy for attacking diabetes and obesity. PTP1B in a different context, its gene is amplified in breast cancer. Overexpression alone is sufficient to cause the disease, and experiments in mouse models have shown that if you cross mouse models that mimic the human disease onto mice that have no PTP1B, you can ameliorate the effects. In other words, an inhibitor of PTP1B would also offer an approach to treatment of this HER2-positive brand of breast cancer. Industry realized this and took approaches to generate inhibitors of PTP1B that were the standard approaches that are taken in industry, where you direct small molecule inhibitors that act at the active site of the enzyme. PTP1B, as all things that are worthwhile, is a challenging enzyme, and what you end up with is ugly looking molecules that have low drug development potential because they're highly charged. So what we really need are new ways to think about how to attack this enzyme. It's highly validated. Industry thinks it's undruggable. This is where academia, people in academia, can think broadly about the problem and come up with different approaches. And we're taking 4 different approaches to inhibiting PTP1B, and I'm just going to focus on this one here, which is we've identified small molecule inhibitors of PTP1B that act by a unique, distinct mechanism targeting sites on the enzyme that are different from the active site, but exert an inhibitory effect at that site. And this is to show you one piece of data. So this is treating a mouse model called the NDL2 mouse, which is a mouse model of HER2-positive breast cancer. It mimics the human disease. Tumours form in the breast and metastasize to the lung, and it's the metastasis that kills you. On the left panel, you see the control sections from the lung, in which you can see metastases, these darker purple spots here. These sections here are sections through the lung of animals that have been treated with the inhibitor at reasonable doses of 5 mg per kilogram or 10 mg per kilogram. And what you can see is that metastasis has been completely abrogated. And it's quantitated over here. These and other data have formed the basis of a strong basis for validating this enzyme as a target in breast cancer and this molecule as an approach to treating that disease. And we're currently going into initiating a clinical trial with North Shore LIJ in breast cancer to try to exploit this further. In the diabetes arena, an essential element of a new drug candidate must be that it is orally bioavailable. Industry has set that hurdle. They want pills that people can take rather than injectables. We have a derivative of this small molecule, and in this experiment what we've done is we've treated animals in what is called a diet-induced obesity model. You feed them the high-fat diet and they get fat. And this is the vehicle control animal that you can see here, and this is its increase in weight. These 2 animals here that have not increased in weight, these are the treated with our PTP1B inhibitor that's administered in an injectable form. But as I have said, the important threshold that we have to get over is oral bioavailability. And these are animals, representative animals, that are treated with this inhibitor orally, and they do not gain weight in this, they lose weight in this diabetes-induced obesity model, same as the injectable format. These are natural products. What we really need, and we've got a focus on these natural product inhibitors, but we would like simpler additional molecules. We have conducted a high throughput screen in collaboration with the Rockefeller University, and although you can't read it here, we've identified 28 additional simpler molecules that show very similar properties, and these could be really next-in-class allosteric inhibitors of PTP1B. And we're in the process now of characterizing these in cell and animal models of breast cancer and diet-induced obesity, and that work is ongoing, following on behind. So the take-home messages. I think we've developed new ways to manipulate this highly validated target that industry views as undruggable, and it just shows the power of the academic approach where you can think broadly about a problem and come up with alternative new ways to think about how to attack it. We've got new drug candidates, including one that's already bioavailable, for multi-billion-dollar markets. If we can cure diabetes and obesity, as I say, we can solve the healthcare properties of the nation. Healthcare problems of the nation. It creates opportunities for commercialization and generating research support, and ultimately, as we get to the more expensive later trials, we hope that the data that we're generating and the new approaches will re-engage industry and get them get them involved in bringing their drug development expertise to bear in the clinical trials. Thanks.
Okay, do we have any questions besides volunteers for taking this pill, right?
Yeah, I'm first.
Larry?
One question I have is, what are the other positive functions of the enzyme PTP1B? Are there any positive?
Its positive function is, it plays a role in a number of different signaling pathways, but its positive function is, as normally as a regulator of insulin signaling, it normally keeps the situation responsive. It's just that in an insulin-resistant state, which is associated with with a variety of causes. Inhibiting this enzyme offers a strategy for promoting insulin signaling and restoring the normal situation.
So in shutting that enzyme off, you're not going to create...
So that's a really good question. No, we're not creating a problem. And actually, the knockout mice are the real validation of that. Those knockout mice have this fantastic phenotype associated with insulin signaling. They don't get cancer. There are no other problems with them. So it is this strategy is a very effective one because there should not be side effects in other systems.
And then just one other follow-up question. What phase of clinical trials are you going into?
Okay.
And how much do you need in funding?
Okay, so the answer to how much do we need is still to be worked out. We're at the stage now where we're waiting for a response from the FDA. This has already been in patients, and it is well tolerated. These were obese patients, and it was very well tolerated. We couldn't get to a maximum tolerated dose. What we want to hear from the FDA is, do we have to repeat that in the cancer patients, or because we have all of these data defining its biological function, can we go straight into a phase 2? We're hoping to be able to go into the phase 2, and we're waiting for the FDA to tell us the answer to that right now.
So tox is not a problem?
No, it's not. It's absolutely. And the effects that we see in the mice are at 20% of the maximum dose that's already been achieved in humans. Yeah.
Any other questions? There's one.
Yeah, talk about your intellectual property patent portfolio.
So there are patents filed around these small molecules. The one that's going into the clinical trial has about 5 years left on that patent. The ones that the orally bioavailable one, there is a much longer patent life associated with that. It's a derivative of this, and its use in diabetes and obesity, its use in breast cancer has not actually been patented yet. So there will be a longer life on that one that has the oral bioavailability properties. And of course, the second-in-class molecules that we have derived through this screen, we own those completely, and there will be patents filed on those at the appropriate time.
Good. One last question.
So I'm a registered dietitian. Are you marketing this as a cure-all, or are you also looking into certain diet factors that help essentially activity?
In terms of so you're asking, would this be a combination therapy, or?
Are you going to look at how diet also accelerates the drug and make it protein?
Yeah, we have not got into that. That would be a good thing to do. I actually thought somebody might ask me this question more in the context of cancer, and actually, but I'm not going to do that. I'll just go to the next one. but there is the possibility of combination therapies, particularly in the cancer context, with some of the existing cancer therapies. We could actually reinvigorate some of these existing cancer drugs that have actually fallen by the wayside. Our data suggests that combinations with those could actually generate new life in those molecules too. But combinations of the sort that you're talking about, we still have to investigate.
Have you applied for any NIH grants?
I've been I have been funded continually by the NIH since October 1990. So yeah, I spent my life writing grants.
August 6th is the closing for the next. That's great.
Thank you very much. That's great. There'll be a line at the back door of volunteers for that clinical study.